Hi everybody, welcome back to this short course of Bioinorganic Chemistry. I have been discussing on the life with oxygen in my last 8 lectures. I have discussed in my previous lectures on how the great oxidizing power of dioxygen is utilized in respiration to produce huge amount of energy. During such respiration process, O2 is fully reduced to water, which would require 4 protons and 4 electrons. However, incomplete reduction of dioxygen produce reactive oxygen species such as superoxide, peroxides and OH radical, which are indeed extremely poisonous for our body. Our mother nature has designed superoxide dismutase, catalase and peroxidase, which not only destroy those reactive oxygen species, but also converts them to something extremely useful for us. I have then discussed about the reaction of dioxin with organic substrates catalyzed by oxygenase enzyme, in which oxygen atoms from dioxygen are incorporated into the final oxidized products that are very useful for our daily life. I also have discussed about various dioxin carrying proteins in biology and highlighted how the beautiful design principle adopted by our mother nature control reversible binding of dioxin in these proteins. Today, I will talk about heme oxygenase, which is indeed responsible for the decomposition of the red blood cells in our own body. We have discussed earlier that hemoglobin and myoglobin are responsible for dioxin transport and storage respectively in our body. In the deoxy form, iron center is 5 coordinate and in high spin state. Once dioxin binds to iron center, oxidation state of iron changes from +2 to +3, while dioxin becomes superoxide O2 minus. This also results in a sharp color change from dull red to bright red. Excess structures of both deoxy and oxy form are shown over here. These two processes are completely reversible, like when dioxin binds to iron center, it forms iron 3 O2 minus, and when it leaves the dioxin, it goes back to its initial state, deoxyhemoglobin or myoglobin. So, this is what is happening, like iron 2 converted to iron 3 O2 minus and comes back to iron 2. And there are of course, a conformational change, as we have already discussed, that high spin iron 2 converted to low spin iron 3 upon dioxin coordination and then goes back. So, during this process, this is what is happening all the time. Now, this dioxin binding slowly causes irreversible oxidation of iron 2 to iron 3 in deoxyhemoglobin. Once they are doing too many cycles, slowly that iron center getting oxidized. This slow oxidation renders iron 3, which no longer can bind dioxin. Once it forms iron 3, it cannot bind a neutral molecule like dioxin. It has to be something like anionic ligand like cyanide, azide, but it cannot bind dioxygen reversibly anymore. So, overall efficiency of hemoglobin decreases over the time. So, there is a need for freshly generated hemoglobin containing iron 2, and what's nature's solution for it? You will be surprised to see that the solution is heme decomposition and regeneration of the heme. Now, question is, where does old heme come from? Mostly is from red blood cell, hemoglobin. As all of us know that red blood cell have a lifespan of only 120 days. So, after that it have to be decomposed, and basically every day it produces around 6 to 8 gram of hemoglobin, and another 10 to 20% of this heme center comes from other heme containing proteins. So, these heme centers actually undergo a decomposition, and nature has designed an enzyme called heme oxygenase in order to do this. So, what is happening? As you can see, that key steps are shown over here, and this converts to heme centers upon removal of globin chains. This heme center actually converts to biliverdin by using oxygen as a oxidant and also NADPH, which is a reducing agent. And during this process, water is being formed, and another very interesting gas, carbon monoxide, is being formed. Please note that carbon monoxide, we have seen earlier, we all know that it is a poisonous gas, but that carbon monoxide also being produced in our body. And this carbon monoxide, although it is a very small concentration, this is very important, and it has been said that this probably responsible for signal transfer in our body. Now, this biliverdin convert to bilirubin, which is yellow in color, which is however insoluble in nature. It have to solubilize first before it can excrete from our body, and for that it conjugate with glucuronic acid and formed a water soluble molecule bilirubin diglucuronide, and thereby it excrete from the body through urine. So, this is what is happening all the time within our body, and heme oxygenase is basically doing the key role in this heme decomposition process. Now, this is how heme undergo decomposition. This heme, in presence of oxygen and NADPH, we call it as a coupled oxidation, because oxygen is oxidant and NADPH is reductant. So, when both used together, it is called coupled oxidation. So, this heme first convert to mesohydroxy heme. You see that there is one hydroxy group which is added at the mesocarbon position, and this molecule is known as mesohydroxy heme. Now, this mesohydroxy heme is highly reactive towards dioxygen. It immediately reacts with dioxygen and convert to heme, as you can see upon liberation of carbon monoxides. And this heme again undergo a ring cleavage exercise in presence of oxygen and NADPH, converts to iron biliverdin complex, and as you can see that ring is being cleaved now. The terminal carbons are converted to keto over here, and then this iron biliverdin complex undergo demetallation, and this converts to biliverdin, which is green in color, and then biliverdin reductase converts to bilirubin. However, during this process there are two things to be noted. The first step is that formation of mesohydroxy heme, which is highly reactive towards oxygen and responsible for its decomposition to biliverdin and bilirubin, and this bilirubin after conjugation with glucuronic acid excretes through the process. Now, another interesting aspects of this reaction is that this iron is getting recycled and reused in heme formations. Please note that there only 2 to 3% of the iron we take every day comes from the diet, and rest 97-98% comes through this recycle process. Still many of us are iron deficient, mostly these women are iron deficient. So, interestingly, as I have said that the heme centers which are very stable, and this has been used extensively by nature in a large variety of enzymes and proteins. For example, catalase, peroxidase, cytochrome P450, hemoglobin, myoglobin, cytochromes, large varieties of enzymes, proteins. However, when it forms this mesohydroxylation, keeping everything intact, it immediately reacts with dioxygen, and the porphyrin ring undergo a cleavage immediately to form bilirubin biliverdin. This is being done by heme oxygenase. As I have just discussed that heme converted to biliverdin, which is not a macrocycle, it is an open chain structure, but highly conjugated, and this is forming out of dioxygen and NADPH, which eventually converts to NADP and produce water, liberates carbon monoxides, and of course, as I have just said that iron is getting recycled during this process. So, this is very important, and this recycle around 98% of this iron which we require regularly in our day-to-day activity, and rest 1 to 2% comes from the diet we take every day. The enzyme which is responsible for this transformation is heme oxygenase, and we find this in spleen and also in liver, kidney, and it requires iron to in heme center to activate dioxygen. This biliverdin, a conjugated open chain structure, it converts to bilirubin, and a biliverdin reductase is responsible for this transformation. So, this double bond is further reduced and CH converted to CH2, and there is a immediate color change. Biliverdin is green and bilirubin becomes yellow. Now, this bilirubin is not water soluble. This is a big problem, and this bilirubin binds to serum albumin, which carries it in the blood. Now, it has to make water soluble, otherwise how it excretes from our body. Bilirubin, after conjugation, glucuronic acid converts to a conjugates, and it becomes now water soluble, and it can excrete through our body. So, if you look at carefully, the red blood cell, which is basically the hemoglobin, after removal of the protein chain globin, this heme unit undergo a cleavage by heme oxygenase with the help of oxygen and NADPH, which eventually liberates carbon monoxide, which supposed to be highly poisonous, but the small amount of carbon monoxide is highly beneficial for our body. It is responsible for various activity. So, during this process, the heme undergo a ring cleavage process, converts to biliverdin, and this biliverdin converts to bilirubin, which is yellow in color, and biliverdin reductase is responsible for this transformation. However, as I have just said that bilirubin is indeed water insoluble. It goes to liver and still water insoluble, and then After conjugation with glucuronic acid, this molecule bilirubin becomes water soluble, and then it goes to intestine, and bilirubin and urobilinogen is formed, which actually reabsorbed into the blood, and also it excrete from our body through urine, and also it deposits on the face. So, you can see the spots on the face, and this is basically stercobilin, and the molecule which excretes in urine is known as urobilinogen. Now, whether you have high level of bilirubin, how you know that? Now, if you go to a doctor, then the doctor will ask you to test the blood to do the two things. One is how much you have the total bilirubin and direct bilirubin. So, from the halus you will get to see that whether you have bilirubin excess in your body. So, actually this blood is getting decomposed to bilirubin, and this bilirubin excretes through the body. This is a spontaneous process, all the time it has been happening. But sometimes if the liver is not very strong, what is happening? There can be more bilirubin in your body, and that creates lots of problem in our body. Particularly this is very common for a newborn baby. Newborn baby mostly get a yellowish tint to their skin, and this is called newborn jaundice. And why this is happening? When the newborn baby take birth, the fetal hemoglobin need to be converted to a adult hemoglobin. So, fetal hemoglobin will convert to adult hemoglobin, and so all the hemoglobin during short period of time will be converting to adult hemoglobin. And if the liver is not very strong, what would happen? That they cannot handle so many bilirubin, and there will be some deposition on the skin, and they develop a disease called jaundice. So, it is very common for the newborn baby, and of course, upon exposure to mild UV light from the sun, it is usually sufficient to destroy this compound. That is the reason in the village you see that mother, they expose their baby to the sunlight. Now, you see that if you have excess bilirubin in your body, it creates a disease called jaundice. You can see that a person having jaundice and a normal person, and you see that his skin color turns yellow, particularly these eyeballs are converted to yellow, and the baby also turns yellow. So, this is very common, and if you go to a doctor, like for the newborn baby jaundice, it is very easy to cure. If you go to a hospital, so what they do, they exposed the kid with blue light. So, it is called blue light treatment. So, you see that the nurse is very happy to see that these kids are getting exposed to blue light, and thereby bilirubin is getting destroyed, and they are also earning lots of money. The nursing home, hospitals, and doctors also earns lots of money out of that, but it is very simple. Of course, there can be some complications, you have to go to the doctor. In the village, mostly the mother, they expose their kid to sunlight, and that UV light is sufficient to destroy this excess bilirubin, which is being deposited on the body skin. Now, going back to this heme degradation process, which is catalyzed by heme oxygenase, as I have just discussed. So, heme center undergo decomposition, first formation of mesohydroxyheme. The heme is dark red in color, and then the vardoheme, then iron biliverdin complex, then After demetallation, it converts to biliverdin, which is green in color, and this biliverdin converts to bilirubin, which is yellow in color. So, action of heme oxygenase can be clearly visible if one see the bruise with time. Say, if you get a fresh bruise, what would happen? It will be red in color. It is happening because of the red blood cell. So, heme oxygenase comes into picture. This red blood cell getting converted to verdoheme, then biliverdin, which is green in color, and then slowly it converts to bilirubin, which is yellowish in color. After several days, bruise completely gets back original skin color with time. So, the action of heme oxygenase can be clearly seen if you follow a bruise changing the color with time, and this bilirubin actually is strongly antioxidant, and thereby it is getting cured automatically. If you have small bruise, you generally need not to visit doctor, since it automatically gets cured due to the formation of bilirubin, which is indeed a natural antioxidant. You do not need to use any ointment or medicine normally. Indeed, this is what is happening in village, where doctors are not easily available, and indeed no medical facilities are there. However, if you go to the doctor will prescribe some ointment which is having some antioxidant, that is indeed doing the same job as bilirubin does. However, bilirubin is a natural antioxidant produced in our body for free, which helps to cure the wound faster. In conclusion, I have discussed today about the heme degradation process catalyzed by heme oxygenase. While oxygen binding is completely reversible in our body, iron 2 centers in deoxy form of hemoglobin and myoglobin also converts to iron 3 slowly with time by oxygen. Thus, the overall efficiency of the protein decreases over time. Nature therefore designed heme oxygenase to recycle the red blood cell, which have lifespan of only 120 days. Bilirubin is formed in the heme degradation process, which is then excreted from our body, while iron getting recycled. This has been a very spontaneous process and happening all the times in our body. However, if there is an imbalance, we will then have several diseases such as jaundice, in which yellow bilirubin is deposited on the skin and the body color turns yellow. In my next two lectures, I will highlight the importance of inorganic elements in medicines. Metal ions play important role in biological processes. You will then see how inorganic chemistry contributes towards such growth. Thank you.